ship it with a protective sticker. For those of you who are used to running a perhaps MinION or Promethean flow cells, the sticker is doing the same job as the valve that we have on the MinION and Promethean flow cells. So we ship it with the sticker in order to make sure that the flow cell doesn't dry out during the shipping process. So it's important to be aware of the protective sticker. The other feature to be aware of the flow cell at this point is on the back of the flow cell. You can see that we have this rectangle here. This is what we call the contact points, and this is the part of the flow cell where we actually connect the flow cell to the flongle adapter itself. It's very important that we establish a good connection between the flow cell and the adapter, and so as a result we ask you not to touch that. We don't want to get any residue on it or anything that might compromise that contact between the actual flow cell itself and the adapter. So the first thing that we need to do is we need to actually load the flow cell into the MinION and the adapter itself. And so for that we need two other components. We need the MinION itself and the flongle adapter. So some of you watching may be fully aware of the MinION and have done some MinION runs before. The MinION, really, there are a few key things to be aware of. Firstly, we have a clip here, which is what we use to actually hold our either MinION flow cells, or in this case, adapter, in place. We also have two holes in the two ports in the MinION, and that helps when it comes to actually loading the adapter into the MinION. And we also have connection ports here, which is also very important when we look to load either the MinION flow cell itself or the flongle adapter. The flongle adapter has a couple of key features as well. On this upper side, you can see the gold rectangle here. These are the contact pads on the flongle adapter, which connect with the contact port on the flongle flow cell itself. So that's where the connection between the adapter and the flow cell itself is made. On the underside of the flongle adapter, we have a couple of key things to bear in mind as well. Firstly, we have the ASIC, which is covered by a thermal tape. The thermal tape is very important to have in place. We need the thermal tape in place because that actually helps to maintain the temperature of the flow cell. It means that we can regulate that temperature through sequencing. We also have two black posts, which we're going to line up with the holes on the MinION, and we have connection pins which line up with the connection ports on the MinION as well. So the first step is to load the adapter into the MinION. The adapter loads in the same way as a MinION flow cell, so slides in under the MinION clip, clicks into place when it pushes to the far end of the, it can't go any further once you've pushed it underneath the clip, and at that point the posts line up with the holes on the MinION and the adapter slots nicely into place. Once the adapter's in place, the final piece is the actual flongle flow cell itself, and the flongle flow cell has got this lip here, which helps us to orientate which side of the flongle flow cell is loaded first. The lip is always loaded first, and in exactly the same way as the adapter, slides in under the MinION clip, slides as far as you can push it, and then all we need to do at that point then is push the flongle into the adapter. You can hear it click. It's now loaded into place. So we've now got the flongle flow cell loaded into the adapter, which is successfully loaded into the MinION. We can now start doing our actual loading. For this, I'm going to place my gloves and goggles on for health and safety. And the first thing that we need to do is remove the protective sticker that we've shipped this flow cell with to avoid it drying out. And as you'll see, as you have a look at the flongle flow cells that you've received, the sticker is only stuck down on one side. There's a loose side here, and that's the side which we use to pull the sticker off. So holding the MinION in one hand and grabbing the sticker with the other, you can start to pull the sticker away from you. Now, initially, you will feel quite a lot of resistance with this because the sticker is held in place by a black bridging port located here on the actual flow cell. And so as you pull the sticker away, initially it's going to feel quite difficult to move away until at this point now, I've now successfully removed the sticker from the bridging ports, and so I can now smoothly move the rest of the sticker, pull the sticker off of the Flongle flow cell and reveal the flow cell itself. At this stage, there are a couple of other key components to the flow cell which are important to be aware of. Firstly, we have a loading port, which is located here. Now, this is the only port that a Flongle flow cell has. So this is the only port that we have to interact with the flow cell. Users of MinION would be used to 2 ports that we interact with. With Flongle, it's just the one. We also then have the array right next to it. That's where the nanopores themselves are located in the membranes. And we also have waste channels, which are located around the outside of the array. And this is where, when we actually flush our sample through, we displace the buffer into these waste channels. So now that I've got the protective sticker open, the next step is to start actually loading the sample. And as I said, for this, I'm going to use pipettes. The first step is to flush a flush buffer, which allows us to displace the yellow storage buffer and allows us to replace it with buffer that we can sequence with. For the purposes of this demonstration, it's just water that we've sent you, but obviously in real life in the lab, this would be flush buffer. And we flush 120 microliters through this flow cell. And so I'm going to take my P200 pipette, set at 120 microliters, take a pipette tip, and all I'm going to do is aspirate 120 microliters of the flush buffer. Now, the key concept to bear in mind here is that we want to avoid introducing any air into the flow cell. And so before you do any flushes into the flow cell, make sure that there is no air at the end of the pipette tip, because we want to make sure that there's no air being flushed into the flow cell itself. And when first-time users on Flongle sometimes struggle to find the loading port, it's located in this indent just at the upper side of the array, and it's right at the bottom of the indent, and you'll know when you're in place because you can actually feel the end of the pipette safely and sort of securely in the loading port. At that point, we want to make sure that the pipette stays at a right angle to the flow cell itself, and then we can, in a fairly slow and methodical fashion, flush through the 120 microliters, which I'm going to start doing now. As I flush through the 120 microliters, you'll hopefully be able to see that we're starting to fill up the waste buffer. So if I give that a quick wiggle, you can see the waste buffer moving through the waste channel. And so we dispense almost all of the 120 microliters. However, the key concept whenever interacting with the flow cell is to make sure that you don't dispense the full volume. Make sure there's a small amount of volume left at the end of your pipette tip. Again, the reasoning for that is we're thinking about making sure we don't introduce any bubbles into the actual flow cell itself. Once we've loaded the 120 microliters and successfully removed the storage buffer from the flow cell, it's now time to actually load our sample. So for Flongle, we load 30 microliters of our sample. So I'm going to take a P100 pipette, set at 030, so at 30 microliters. And I'm going to take my loading beads. Now, anybody who's ran an experiment on a sequencing run on MinION or Promethean will be fully aware that the loading beads that we add towards the end of our sample prep do sediment very quickly. So it's always just worth making sure that you give this a bit of a mix by inversion to ensure that the loading beads are homogeneous and nicely suspended in the sample. Once you're happy with that, it's a case of taking 30 microliters of your loading beads. Again, make sure there's no air at the end of your pipette tip. Place in the loading port. Once again, once it's in place, it should feel quite sturdy. It's not going to slip around anywhere. If you are at all unsure, whether you've got it in the port or not. Dispense a small amount. It'll become very clear if it's not, because either the sample or the buffer will start pooling on the surface of the flow cell. So it'll be very clear if that's not the case. Again, 90 degrees angle, and I'm going to start dispensing my 30 microlitres. And as I dispense, I'll keep an eye on how much I've got left. And towards the end, I keep the pressure on my pipette tip, on my plunger, remove my pipette, and as you can see, again, I've left a small amount of liquid in the actual pipette tip itself, so that I'm not introducing any air into the flow cell. Once that's done, we've now loaded our sample. That flongle is good to go. The last bit is just to close it up and to actually ensure it's ready for a sequencing run. And in order to do that, we're going to replace the sticker. Now, as we replace the sticker, the important thing to bear in mind is to do this by a dabbing motion, rather than don't rub it, a dabbing motion, and ensure that there's a real nice seal around that loading port, so that during the sequencing run, the loading port isn't going to dry out. We can then close the MinION, and that would then be ready for sequencing. So that is a complete flow cell ready to sequence. So I hope you found that part of the demonstration useful. I'm going to take my goggles off for a second. I hope you found that part of the demonstration useful. Now it's the opportunity for you guys to do this alongside me. So before we do this, just a slide on the key points to remember. We've got 5 key points. When removing that sticker to begin with, try and do it in as smooth a fashion as possible, being aware that there's going to be that initial resistance from underneath the tab. When you are first loading either your sample or doing your flush, ensure that you aren't introducing any air by having any air at the end of your pipette tip. When you're doing a flush, don't dispense everything. Keep a small amount of volume in the end of your pipette tip so that you don't introduce any air. Replacing the sticker is in a dabbing fashion rather than rubbing, and ensure that that loading port is really nicely sealed. So I'm going to get myself sorted here and get ready for the next part of the demonstration. While I'm doing that, if you guys can now open up your foil pouches, in there you should find 2 flow cells, a syringe with the syringe tips, and 2 Eppendorfs, one labelled as flush buffer and the other labelled as loading beads. So if you can do that now, I'm just going to get myself sorted and we can go through that part of the demonstration. So hopefully everybody has got that and we are now good to go. I'm now going to put my goggles back on. If I could ask that everybody at home, you do the same, so that we're all complying with health and safety, then we'll start the demonstration. So hopefully everyone's at the point where they've unpacked their pouch, they've got the flow cells, syringe and the 2 Eppendorfs, and have got gloves and goggles on. And if that's the case, then we will proceed. So with the constraints for this year, unfortunately, we haven't been able to send out flongle adapters to you as well. And so we're going to have to make do with loading the flow cell independent of the adapter. But that shouldn't be a problem. It's still fairly straightforward to do. And so we should still get the real feel of how this is going to work. So as I highlighted in the demonstration earlier, the first step with all of this is to make sure that that we remove the protective sticker. So if everybody can do this at home alongside me. So we're going to hold the flow cell in one hand between the thumb and the first finger. You can hold it, just to make sure it's in place on the table. And with your other hand, lift off the part of the sticker which isn't stuck down. And then, as I showed earlier, it's simply a case of lifting that sticker up and then pulling away from you. And as you do that, hopefully you'll feel what I was talking about earlier in terms of the sort of the pullback from the initial, from the bridging piece. But as you move past the bridging piece and you successfully remove the sticker from the bridging piece, it then becomes much easier to unpeel the rest of the sticker from the flow cell. And as you do that, you will open up the loading ports. Now, I have had some people, when they've done this, pull quite hard to the point where they've pulled the sticker off of the flow cell, and that often causes a bit of panic. There's absolutely nothing to worry about if that happens. You haven't broken the flow cell will work entirely as it should do. There's nothing to worry about with that. All I would say is make sure you do remember to keep hold of that sticker, because as I showed earlier, we need that sticker to be replaced once we finish loading in order to cover the loading port and make sure that that flow cell doesn't dry out anymore. So with the sticker removed, and now we have access to the loading port, now we can start to do the flushes. And the first flush that we need to do is we need to flush the flush buffer with 120 microliters through the flow cell in order to displace the yellow storage buffer that we've shipped the flow cells in. And so with the syringe, where we want to get to is the second notch after the 0.1 measure on the syringe. So that will be 120 microliters. And taking the syringe and the syringe tip and open up the Eppendorf, we can now aspirate the syringe until we hit that 120 microliter mark. And once we've got the buffer in the syringe tip, we can then place into the flow cell. Now, again, the key concept here is make sure that you don't have any bubbles, any air at the end of your syringe tip. Perhaps a little bit more difficult to see here than with the, well, certainly more difficult to see here than with the clear pipette tips, but hopefully you can see what the concept is for this. And then this is the first time that you'll interact with the loading port. As I mentioned before, it's on an indent, right at the bottom of an indent in this flow cell. And so now's a good time to really orientate yourself with where the loading port is and really start to get a feel for what it feels like to make sure that your syringe tip and therefore your pipette tip, when you're running this in the lab, how that feels when it's successfully in the port. Once you're happy with that and the syringe can no longer slide around, then make sure that you can maintain this 90-degree angle between the syringe and the flow cell itself. And then it's a slow, gentle dispense of the 120 microliters. Again, it's very important to be aware that we're not going to dispense all of this buffer. We are going to there we go. We are going to maintain some buffer in the end of our pipette tip. Reason being is that we don't want to introduce any air into the flow cell once we've loaded. So obviously at this point in the lab you would replace your pipette tip with a different pipette tip. However, because we've only got one syringe, I'm just going to replace the remainder of the flush buffer that I have in my syringe back into the flush buffer Eppendorf. So hopefully everybody is at that stage. You've had an opportunity to really feel what it's like when the syringe tip is successfully located in that loading port, and hopefully you've had a chance to feel how that feels if it isn't quite in place. Obviously, if you're ever concerned, dispense a small amount of buffer to begin with. If it's not in place, then you'll quickly see a pool of buffer appearing on the surface of the flow cell. At that point, you're going to be well aware that something isn't quite right in terms of where you're loading. So I'm just going to give everyone just a couple more seconds to make sure that we're all up to speed and everybody has loaded that 120 microliters and successfully displaced the yellow storage buffer from the array. So hopefully we've all done that, and we can all now move on to the next step, which is the actual step of loading our sample. So this is using the aliquot, the Eppendorf labelled as loading beads. Again, this is just water for the purposes of this, but in the lab this would be your actual sample. And as I mentioned, we want to make sure that the loading beads are nicely homogeneous within the Eppendorf. So mix a little bit by inversion just to make sure that the beads are well suspended. And we're wanting to load 30 microliters at this stage. On the syringe, this is a little bit more tricky because it's literally the third notch up on the syringe. So there isn't, you haven't got much volume to play with here. It's just the third notch up. And again, place your syringe tip in the loading buffer, in the loading beads, apologies, and load your whole sample up to the third notch. Once you're up to the third notch, ensure that you haven't got any air in the end of your tip, which actually I have. And maybe you can see that there are different, slightly lighter rings. That means that I've got a little bit of air in my in the end of my syringe. So I'm just going to dispense some out, and hopefully now you can see that that's looking a lot more homogeneous. And then again, I'm going to place in the loading port, again making sure that there's no movement, no slipping around, and then dispense my 30 microliters. Now, again, ensure that you don't dispense everything. Make sure you've got a small amount left in the tip of your syringe. Again, the key concept of that is making sure that we're not introducing any air into the system. Now, you might notice that I've got a small amount of buffer coming out of one of my waste ports. That's perfectly fine. That can be, that we expect that to happen. That's not an issue. If you do see that, then just clearing away with a little bit of dabbing away with blue roll or anything else that you have in the lab, just to remove that would be absolutely fine. And then the final step, so just make sure that everybody's caught up. So if everybody has a few more seconds to do that final load of 30 seconds before we move to the final step of replacing the sticker. Okay, so hopefully everybody has done that. We've loaded that 30 microliters of our sample, and the final step is to replace the sticker. Now, as I said, if the sticker's been ripped off, that isn't the end of the world. All we have to do is make sure we successfully replace the sticker back on the Flowcell, with the key point being making sure that that loading port is nicely sealed. So I'm going to do that now, and take the sticker and dab in a dabbing motion. I'm going to add the sticker, so not the rubbing. I want to dab into position, and again, making nice and clear that the loading port is well sealed, so that when we come to doing the actual sequencing run, the flow cell won't dry out. Okay, so I'm going to take my glasses off because that's the end of the demonstration. At this point now, we have a fully loaded Flongle flow cell ready for sequencing. The final step here would then be to close the MinION lid, because in the lab this will be connected with the Flongle adapter, which is loaded into the MinION. So we would close the MinION lid, and we would then be ready to start sequencing. So just a quick reminder of what those 5 key points are to bear in mind when loading a Flongle flow cell. Ensure that you remove the sticker in a smooth motion, bearing in mind the fact that there will be a little bit more resistance to begin with as you start to remove the sticker from underneath that bridging piece. Once you're loading, ensure that before you load that there is no buffer at the end of your pipette tip. Similarly, towards the end of loading, make sure that you don't dispense the whole volume into the flow cell. Make sure that you leave a small amount of buffer or sample at the end of your pipette tip to make sure that you're not introducing any air into the flow cell at any point. And then when it comes to re-stickering the flow cell, make sure that you're applying in a dabbing fashion, and similarly that the loading port is nicely sealed. So I'm going to leave it there. As I mentioned before, there will be a Q&A session right after this. I'd be more than happy to answer any questions that you may have about Flongle or flow cell loading in general. So please feel free to submit your questions to that, and I look forward to discussing flow cell loading in a little bit more detail then.